Class, 2 participants I can see is raising hand. Yes, आज पर्व को बोलने देते हैं. चलो पर्व बोलो.
Yesterday, ma'am, we studied about nucleus, chromosome, types of chromosome.
So in chromosomal structure, a few more important parts I wish to say, and then at last we will see a few one video specifically I wish to share with you, which will give you idea the parallelism of DNA, chromatin reticulum, chromosome, and gene also, which is very important for us to know. See, if you look into structure of a chromosome, you can see that 2 chromosomes are connected with each other by means of, 2 chromatids in a chromosomes are connected with each other.
Centromere.
Centromere. And this is one chromatin. So each chromosome will have 2 chromatids. The chromatids of same chromosomes are known as sister chromatids.
Sisters.
But if we have 2 chromosomes present with us, these 2 chromatids are known as non-sister chromatids. Now, what are they actually? If you closely look into structure of chromosome, each chromosome are having various attributes that I am specifically wish to discuss today. In chromosome, if you look closely into structure of a chromosome, the best time to study structure of chromosome is the metaphase state of cell division. So the metaphase state of cell division is the best time to study chromosome. Why? Because at metaphase state, cells are about to just divide the genetic material. उसके जो genetic material है, it is just about to divide into 2 different parts. क्योंकि कल हमने डिस्कस किया था कि at the time of cell division we require them to coil. So that is why at this stage we can clearly see the chromosomes, and there is a procedure to study chromosome that is called karyotype. Yesterday it was mentioned in the video, but I am mentioning it in case if anyone missed it. Karyotype means study the structure of chromosomes. There is another name or terminology we need to talk about, that is called idiogram. What do we mean by idiogram? It is the diagrammatic representation of chromosomes. So I want to show you one idiogram. Let me share the screen. So consider, if I am talking about chromosome of any organisms, wait for a moment. Is the screen visible, beta?
Yes, ma'am.
So you can see If any organism, this is in case of human being, I am talking about this one. Let me use pointers. यहाँ पे 46 chromosomes है because for a single species this chromosome number is conserved. In normal situation human के पास हमेशा हमेशा 46 chromosomes ही रहेगा. It can never be less or more than that. हाँ, if a person is suffering from any disorder which is genetic disorder, this number may vary. तो आप देख सकते हो यहाँ जो chromosomes है, it is representing a set of chromosome. मतलब हमारे पास जो 46 है उसमें से एक सेट को ये रिप्रेजेंट कर रहा है. That is why 22 pairs of chromosomes हमारे पास है. तो 1, 2, 3, 4, 5, 6 करके it is always representing up to 22, and then it is expressing the X and Y chromosome. वो अपना X chromosome और Y chromosome दोनों को represent कर रहा है. अब आप पूछोगे, you may ask me question कि मैम क्यों 22 को represent कर रहा है? Why it is not representing all 46 chromosomes? क्योंकि बेटा chromosomes exist in pair. ये फर्स्ट वाला क्रोमोसोम जैसा है, इसका ऑपोजिट भी वैसा ही देखने में होने वाला है. If you look into the structure of the other chromosome, it will be same as the structure. इसको हम बोलते हैं, इनको homologous chromosomes. इनमें से एक हमें अपने फादर से मिलता है और दूसरा हमें अपने मदर से मिलता है. तो कंसीडर करो एक ऑर्गेनिज्म में 10 क्रोमोसोम्स हैं, तो उसमें से कितने क्रोमोसोम्स हमें उस ऑर्गेनिज्म को उसके मदर से मिला होगा? Out of the 10 chromosomes, if a person or organism is having, उसमें से कितने उसको उसके मदर से मिला होगा बेटा?
5.
Yeah, yes Ayushi, 5. 5. So और 5 मिला होगा फादर से. तो अब अगर इनको आप मैच करोगे तो देखोगे फर्स्ट पेयर ऑफ क्रोमोसोम जो है, दे हैव सेम स्ट्रक्चर. सेकंड पेयर विल हैव सेम स्ट्रक्चर. थर्ड पेयर विल हैव सेम स्ट्रक्चर. तो दीज क्रोमोसोम्स आर नोन एज होमोलोगस क्रोमोसोम. ओके, होमोलोगस क्यों बोलते हैं? बिकॉज दे हैव स्ट्रक्चरल होमोलॉजी और सिमिलरिटी. सो दिस इज व्हाट इज इडियोग्राम. So idiogram is known as diagrammatic representation of chromosome. क्योंकि chromosomes को आप अगर actually देखोगे, so they are very difficult to study. So obviously we need to diagrammatically represent them. What are the other things we need to know? We need to know about the constrictions of chromosome. If you look into this depression present in the chromosome, this is the part where centromere is present. This is known as primary constriction. It is known as primary constriction. Okay. दूसरी वाली किसी केस में, in some specific cases, in some chromosome there is another depression observed. If it is observed, that type of chromosome is known as satellite chromosome. क्यों satellite? इसकी एंड दूर से देखने में सैटेलाइट जैसी दिखती है. That's why it's known as satellite chromosome. So in that case, this will be the primary constriction. So let me label it this way. It will be easy for you to understand that this will be the primary constriction, and this constriction which is giving it a small head-like structure like this is the satellite structure, is known as secondary constriction. Yesterday we discussed about the types of chromosome, and in that video the types were described very briefly. They talked that we need to measure the size of these arms. तो दोनों आर्म में से जो छोटा होगा उसको क्या लेंगे और जो बड़ा होगा उसको क्या लेने वाले हैं? Yes Ayushi?
Ma'am,
one person at a time please.
Ma'am, longer head is known as Q arm and shorter is P arm.
P and Q. सो ऑब्वियसली हम P और Q का रेशियो निकालेंगे. अगर P by Q का जो रेशियो होगा, और rather to say sorry, Q by P का जो रेशियो होगा, if it is 1, we call them सब metacentric. मतलब इनके केस में जो सेंट्रोमियर है वो एकदम बीचों बीच प्रेजेंट है. इफ दिस Q by P का जो रेशियो है that is greater than 1, it can never be greater than 1 actually, as we took this. If this is taking less than 1, we call them submetacentric. मतलब उनके केस में कैसा होगा? The centromere is not located exactly in the middle. But if this value, the Q by P value is nearly 0, then we call it acrocentric. मतलब उसके केस में कैसा होगा? It will be a specific chromosome where centromere is located at the very end of the chromosome. Understood? This part of types of chromosome is clear to everyone. So this is metacentric, meta. This is submetacentric. Okay. Then there are acrocentric, and this one is telocentric. So various numbers are there which vary from species to species. That number will decide. एक standard number होता है, they give it to you. By referring that number, with finding this ratio, you can understand the types of chromosomes. Now, let us. This is all about structural details of chromosome. I just want to ask you. Yes, बेटा, बोलो.
मैम टेलोसेंट्रिक एक बार फिर से उसका स्ट्रक्चर बना के दिखाओ ना.
हाँ, बना तो हुआ ही है. I can show you. This one is telocentric. Actually, it is very important to understand the difference between submeta. See, if structure is like this, it is submetacentric. If it is like this, it will be acrocentric. And if it is like this, then it will be telocentric. So this is submetacentric, this is acrocentric, this is telocentric. Simply the ratio will decide the structure. Clear?
Yes, ma'am.
So now we will talk about chemical composition of chromosomes. Chromosomes are made up of what? Chromosomes are made up of structures which are actually made up of proteins and DNA. And very trace amount also the RNA is also present. Now obviously yesterday I made you understand that chromatic reticulum or DNA coils around histone proteins to form chromosomal structure. Now at the centromere position, what is exactly present? The centromere is made up of two disc-like structures like this, which are connected by interconnected spokes like this. This shaped structure is known as, so if you look into chromosome, this is how they look. We call them kinetochore. What we call them? Kinetochore. And kinetochore is the area जहाँ पे cell division के time पे the fiber which is stretching and separating chromosomes into two half, it is the kinetochore, the area where it is getting attached. The fiber which is stretching and separating chromosomes into two equal halves will be attached to the kinetochore. Any doubt about any structure of chromosome and everything?
No, ma'am, no doubt.
Can you repeat this?
Yes, Parth, what you want to say, beta?
And just to study, can you repeat? I have network issue, therefore I don't listen it.
No problem. I can repeat. I'm sharing the screen again. You let me know which part. You missed the part called kinetochore?
Yes, ma'am.
So basically nothing. If you have a close look into the centromere area, you can see two disc-shaped structures which are actually connected with each other with the help of spoke-like structures. This plate-like structure is called kinetochore, where actually at the time of cell division, the fibers bind and then they what I can say, then they drag chromosomes into two different parts. सेल के दो पोल में वो क्रोमोसोम को डिवाइड करके देते हैं, है ना? कंसीडर करो वन सेल से टू सेल बनते हैं, सो सेल में जो भी चीजें हैं इट शुड बी इक्वली डिवाइडेड इनटू टू पार्ट्स. नो, we will discuss about it in detail in cell division chapter. That's all. So now I wish to again share one video. It will be a very very important video. Why? Because it will talk about the relationship of DNA, chromatin reticulum, chromosome as well as gene. What do we mean by gene? So I want everyone to look into it and note down if you at any point if you have any doubt. And with this we will be done with the chapter, this chapter, that is cell. So then we will once again revise the part and we can take your doubts. I am sharing the screen for the video. Is the screen visible, Rita?
Yes, ma'am.
Yes, ma'am.
Visible.
So let me start playing it.
What if we get to know the probability?
I guess the sound is audible to you clearly, beta?
Yes,
No, ma'am.
ma'am.
Yeah, na?
No, ma'am.
No, ma'am.
Of us falling a prey to a particular disorder in advance?
Is it?
What if we can predict serious disorders like cancer using simple medical testing? Is that possible? Or is it just fiction?
Is it audible now?
Yes,
Yes,
ma'am.
ma'am.
So let's
Yes,
start
ma'am.
from the beginning.
What if we get to know the probability of us falling a prey to a particular disorder in advance? What if we can predict serious disorders like cancer using simple medical testing? Is that possible? Or is it just fiction? Well, no. It's already being done. The concept of genetic testing, or the technique in which our genes or chromosomes are tested, help us here. So what are chromosomes? We've always come across the saying, humans have 23 pairs of chromosomes. But what are they made up of? And where are they actually located in the cell? Let's try and answer all of these queries in this session. Here we have this simple cell as an example. Can you see the thread-like structures here in the nucleus? These threads are called chromatids. They are usually tangled in this fashion when present in a regular active cell. Now, what if this cell has to undergo division? Is it possible to divide this single cell into two equal halves with such a tangled bundle of threads? Of course not. Imagine we have these different colored threads tangled together. If we were to separate them, then what should be the first thing we should do? We will first separate each thread and then make separate bunches. In a similar way, even the cells separate these chromatid threads. This process is aided by condensation of the threads. It means these chromatid material will gradually start coiling like a telephone wire. As a result, the threads will gain structures like these. Are we familiar with this kind of a structure? Have we come across it before? That's correct. It is the structure of a typical chromosome. What are chromosomes? Well, we'll get into their details in the upcoming sessions. For now, we need to get a basic idea about them. So we have this cell with threads sorted in this classical chromosome structure. This structure is seen only when the cell is in the process of division. So can we say that both chromatids and chromosomes are the same? Yes, they are. The different names are for the different structures. Now the next question is, what are these threads, or chromosomes to be precise, made up of? If we zoom into the structure of this chromosome, we find small segments like these that make up the complete structure. These functional segments are nothing but genes. So can we say that chromosomes are made up of genes? Absolutely. All these segments make up the whole structure. Now let me ask you one more interesting question. Do you think the genes will also be made up of something? Will there be units that make up these segments? If your answer is yes, then you're absolutely right. Genes are also made up of some units. If we zoom inside this gene segment, then we find large molecules like these that make up the segment. These large molecules, or macromolecules to be precise, are DNA molecules. This is not a new term for us. We are quite familiar with the term DNA as it's very commonly used these days. But what exactly are these DNA molecules? Well, this single large molecule is composed of 3 units. And what units are these? Let's reserve this explanation for our upcoming videos. For now, all we need to know is that DNA is made up of these units. Now to quickly review what we learnt, let me remove the text from the screen. These 3 combine to give us a single molecule. And many such DNA molecules form long chains. What is a sequence of DNA molecules called? That's right, it's called a gene. So technically, segments of DNA molecules which form functional units are called genes. And many genes come together to form thread-like structures called chromosomes. Chromosome is the classical structure obtained only during cell division. When the cell is not in the dividing phase, the genetic material is present in the form of tangled threads called chromatids. So this is the basic idea of the genetic materials in our cells, which we needed to understand in order to look at the laws in genetics. Let's get introduced to the laws in genetics put forth by Gregor Mendel in the next part.
So class, wasn't the video very nice one?
Yes, ma'am.
Yes.
Yes, ma'am.
Explained very well the concept of parallelism between gene and chromosome. So actually they talked about gene beta, but we cannot see genes. When we look into the gene, अगर हम chromosome में देखें under microscope, genes are not visible because there is no demarcation of gene. कि अच्छा यहाँ से यहाँ ये जीन है, यहाँ से यहाँ तक ये जीन है. जैसे आज हमने जब इडियोग्राम डायग्राम देखा था, 1 मिनट विकी, मैं एक बात बोल दूं बस. आई एम कमिंग टू यू. डायग्राम हमने जब देखा था उसमें ऐसे लाइंस किए हुए थे. इफ यू कैन रिमेम्बर, those lines were representing gene, but हम जब chromosomes को माइक्रोस्कोप के नीचे देखते हैं तब हमें genes नहीं दिखता. But then why and how these lines are there? For our benefit, हम क्या करते हैं, क्रोमोसोम के पार्ट्स को इमेजिनरी लाइन से डिवाइड करके एक डायग्राम से रिप्रेजेंट करते हैं. जैसे डू यू थिंक इक्वेटर एग्जिस्ट? I hope all of you.
No, ma'am.
Hello?
There is an imaginary line.
No, ma'am, it is imaginary line.
वहां किसी ने लाइन खींचा नहीं. But for our benefit or study the structure of Earth and about geographical location and everything, we draw these imaginary line, equator, Tropic of Cancer, Capricorn, then latitude, longitude. Oh, it is for us. No such line is there. Same is for genes. They exist. They exist in their function, not exactly they are lined up in the chromosome. What is their function? They code for protein, which ultimately decides the character, of organism. Yes, Vicky, what was your doubt? Tell me. बोलो बेटा. Vicky, you wanted to say something, beta?
Yes, ma'am. Yes, ma'am.
Bolo.
Ma'am, why we cannot see the genes under microscope?
Because there is, when we see anything under microscope, it should be stained separately, no? It should be stained separately. हम माइक्रोस्कोप में अगर प्लेन एक सेल को देखेंगे, it will be colorless because their cells are transparent, they are very small, है ना? तो उसको देखने के लिए what we do? I taught you microscopy, you know, we stain them. Okay?
Yes, ma'am.
So by staining method, we can locate कि अच्छा यहाँ से यहाँ इतने portion में genes are located. There are techniques which are called dish and fish. मजेदार names है. Fluorescence in situ hybridization technique is there, by which we can definitely have an idea about gene. But normally जब हम chromosomes को देखते हैं microscope के नीचे, chromosomes itself are very small. I can show you some slides of chromosomes images I can show you. हम जैसे imagine करते हैं, they don't look like that. Let me share, you will have an idea. Can you see different chromosomes?
Yes, ma'am.
Yes, ma'am.
I prefer to show directly in the Google because you can see them many. अब देखो, certain chromosomes are very big. For example, in this case, you can definitely understand the structure of chromosome. एकदम साफ दिख रहा है कि बड़े बड़े क्रोमोसोम्स के आर्म हैं, chromatids are there, isn't it? But see, they are smaller in size. And try and look human chromosome. Can you identify them? So small they are.
No, ma'am.
No.
Why this happens? Because more the number of chromosomes in an organism, जितने number of chromosomes बढ़ जाते हैं, उतनी कम जगह में accommodate करने के लिए they have smaller sizes. So that is why when you naturally look into look into them under microscope, you can see that there is no such thing called what I can say genes visible under chromosome. For our benefit, we created because we cannot stain them separately like that. Anything else, anyone?
Ma'am,
No, ma'am. No doubt.
हाँ बोलो बेटा.
ma'am, what are primary constrictions and what are secondary constrictions?
Primary constriction and secondary constriction. These are two depressions I described, no beta. Primary constriction is the area where centromere is there, and secondary constriction is the area actually, let me tell you, there is a chromosome, specific chromosome. I told you about nucleolus yesterday, that at the time of cell division, nucleoli is attached to the secondary constriction of specific chromosomes. Clear? Class?
Yes, ma'am.
So let me know how you wish to revise the concept of cell today. I request all of you to do one thing better. Class, am I audible and you can see me?
Yes, ma'am.
Yes.
Yes, ma'am.
Ayushi, can you please respond if you can hear my voice or not?
Ma'am, I can hear you.
Okay. I think some problem was there because I cannot hear any one of you. So it is approximately 6 feet 2 meters long. Class, am I audible?
Yes, ma'am.
So it is 6 feet 2 meters long. Next doubt. Class, I just want to ask if you have any doubt, beta. Do you have any doubt? Anyone? I cannot hear any one of you.
No doubt, ma'am.
No doubt.
So then how to revise? Yes, Parv, you want to say something, beta? So let us have a very quick quiz round and let me check how much you know about the prokaryotes and eukaryotes. You need to raise your hand to give me the answer of the questions. Are you ready to play a quiz? No one will speak without raising hand. Can we?
Yes, ma'am.
We will have quiz on Monday. All right, Yashvi?
Okay, ma'am.
So we will meet on Monday and we will discuss a lot and we will have a quiz game. Is anyone having any doubt?
No.
Class? Okay then. I am ending the meeting. Bye bye to all of you. Good night.
Yeah,
Bye,
ma'am.
ma'am.
Bye, ma'am.
Good night. Good night.